Welcome to another deep dive, everyone. Today we're going to be talking about anacondas. Yeah,
Anacondas.
specifically the green anaconda. We're going to be looking at a paper that was published last August 2024.
Oh, wow. So pretty recently.
Yeah, just a few months ago, called How Not to Describe a Species, Lessons from a Tangle of Anacondas. And this was
Catchy title.
yeah, and it sparked a lot of discussion in the taxonomy community. So we thought it'd be interesting to kind of dive into this and
Yeah, like go behind the scenes a little bit of what.
yeah, and figure out like what happened here. And so for folks who maybe aren't super familiar.
Yeah, let's set the stage.
Yeah, so there are 4 generally recognized species of anaconda, and of course the green anaconda is the biggest one that everybody knows about.
Right, the one we all think of.
Right, and so this paper basically tried to say like, hey, you know what? There's actually like a 5th species of green anaconda that
Yeah.
we're going to call Eunectes acaima, and a lot of people in the scientific community were not
Not having it.
having it. So
So what went wrong?
yeah, like
Yeah.
what's the deal here?
Yeah.
So so one of the things that we noticed is that this paper relied really heavily on mitochondrial DNA,
Okay, yeah. And that's a tool that can be useful, but it can also be really misleading.
especially by itself, right?
Yeah, it's like if you're trying to understand the history of a town, but you only look at the records from one family, you're going to miss a lot of stuff.
Yeah, you're not getting the whole picture.
Right, exactly.
Yeah.
And so mitochondrial DNA is only telling you part of the story because it's only reflecting the lineage of the mother.
Oh, okay. Interesting.
And it can be affected by things like, interbreeding between species and all kinds of different.
So even if there are differences in the mitochondrial DNA, it doesn't necessarily mean it's a new species.
Exactly right. You need a much more holistic approach.
Yeah.
If you're going to say, hey, this is a new species,
Okay.
you need to look at nuclear DNA,
Okay.
which gives you a much more complete picture of inheritance. And you need to look at morphology, the physical characteristic.
Well, hold on now, because they said in this paper that they did look at nuclear DNA.
They mentioned it,
Yeah.
but they didn't actually publish the data.
Oh, so they just said they did it.
I said they did it,
Okay.
which, when you're making these kind of claims and there's already controversy,
Raises some red flags.
that raises a red flag for sure.
Yeah. Okay, so what about the physical characteristics then? Were they able to find anything there that sort of
They actually said that there were no significant morphological differences,
Okay.
that they weren't notable,
Okay.
but other scientists have come out since then and said, like, no, that's not true.
Oh, really?
Like, if you look at the data, there's actually variation
Okay.
that you can see between green anaconda populations that contradict that.
Interesting.
So that's another thing that makes this paper a little
shaky.
shaky.
Yeah. Okay, so if they didn't really do their due diligence with all that other stuff, then what made them think that they had a new species? Like, what did they find in the mitochondrial DNA that...
Yeah, what was so compelling?
Yeah.
Well, they focused on a particular mitochondrial gene called CPAB,
Okay.
and they found a 5.2% difference
Between the...
between the proposed new species and the existing green anacondas.
Okay.
And they said, okay, that's different enough that this must be a new species.
5.2%. That seems like a lot
It sounds like a lot.
in terms of like genetics and stuff. I don't know.
Yeah.
Is that...
It's not that simple.
It's not that simple.
You
Okay.
have snakes that have less than a 5% difference in that gene that are very clearly different species.
Oh, wow.
And you can have snakes that have more than a 5% difference that are the same species.
Okay.
So it really
So
depends.
you have to look at everything,
You have to look at everything.
not just this one little piece.
Exactly. And it seems like they were really focused on that one piece of data.
Okay, so they kind of jumped the gun, maybe?
Yeah. It seems like they may have jumped the gun.
Okay.
And it's not just the scientific method that was a little iffy. The naming also kind of went off the rails.
Oh, no. What happened?
They broke a bunch of rules.
With the naming?
Yeah, they broke some rules in the International Code of Zoological Nomenclature.
Oh, there's like a rule book
There's a rule book
for naming stuff.
for naming stuff,
Oh,
which makes sense.
yeah.
You need some rules.
Okay, so what did they do?
For example, they wanted to use an indigenous name for the species,
Okay.
Acaima.
Okay.
And they said, well, this name has been used for much longer, so it should take precedence over any scientific names.
Oh, so they were trying to make their own rules, too.
Yeah,
Okay.
but the code says that published scientific names are what matters,
Okay.
because you need to have that like universal system.
Yeah, that makes sense. You can't
Yeah.
just have everybody calling it whatever they want.
Right, exactly.
Okay, so what about the name Acaima itself? Is that even like a valid scientific name?
Well, that's another problem. It turns out that the way the paper was published online might mean that that name isn't even valid.
Oh, wow.
And even if it was valid, there are actually other scientific names that would take precedence because they're older.
Oh, they didn't do their research very well.
Seems like they missed a few steps.
Yeah, okay. So we've got like iffy methods, missing data, naming errors. Is there anything in this paper that like holds up?
So actually there's kind of a fascinating twist to this whole story.
Okay.
The original reference specimen for the green anaconda, it's called the lectotype.
The whatotype?
The lectotypes.
Okay.
It was thought to be lost.
Oh.
But guess what?
They found it.
They found it
What?
in a Russian museum.
A Russian museum. What was it doing in Russia?
Well, that's a story for part 2.
Oh, come on.
But I'll give you a little hint. It involves a journey back in time,
Ooh, I like it.
a famous naturalist collection,
Okay.
and some surprising connections that link scientific discoveries across centuries.
Okay, well, now I really want to know.
Stay tuned.
I'm hooked.
All right.
Oh,
Okay, so are you ready to go back in time?
lead the way.
All right, so this lost lectotype, it takes us all the way back to the 18th century,
Wow. Okay.
to this incredible collection of a Dutch naturalist named Albertus Seba.
Sabo, wait a minute. Wasn't he like the guy who had basically his own personal zoo and botanical garden before those were even really a thing?
You got it.
I think I've heard of this guy.
Yeah, he was a pharmacist by trade, but man, he loved nature.
Okay.
He was obsessed with it. He amassed this huge collection of plants and animals from all over the world, and his home in Amsterdam was basically like this cabinet of curiosities
Oh, that's cool.
that people would come from all over to see.
So the green anaconda lectotype was part of his collection.
He included a description and an illustration of this particular anaconda in his book, Thesaurus,
Oh, wow.
which is basically like an encyclopedia of natural history. It was a huge deal back then.
Yeah. Okay, so we have this VIP specimen. It's described, it's illustrated in this landmark work, and then it just vanishes. How did it end up in Russia?
Well, after Seba died, his collection was sold off in pieces.
Okay.
And some of those specimens, including this anaconda, ended up being bought by Catherine the Great.
No way. Catherine the Great, the Empress of Russia, are we talking about?
That's her, the one and only.
The one with the crazy art collection and the even crazier personal life that Catherine the Great
That's
had.
the one. She was a big supporter of the arts and sciences, and she wanted to expand the collections of the new Russian Academy of Sciences.
I see.
And Seba specimens were perfect for that.
Yeah, they added a little like exotic flair.
Exactly, a little prestige.
Okay, so this anaconda went from a Dutch pharmacist's like personal museum to the hands of an empress. That's quite a journey.
That was quite a journey.
But how did it go unnoticed for so long? Were scientists just not aware that it existed?
Well, it seems like there was a bit of a mix-up over time. The records got kind of messed up,
Okay.
and they lost track of the anaconda's connection to Seba's original description.
Oh, so it was like right there under their noses?
They knew there was a lectotype out there somewhere. They just couldn't find it.
Like a scientific cold case.
Exactly. And then, boom, rediscovered in Russia.
That's got to be pretty exciting for the researchers who found it.
Oh, yeah. It was a big deal. It just shows you how important these museum collections are.
Yeah.
They're not just displays. They're like living archives.
Yeah.
Full of clues that can help us solve these scientific mysteries.
Wow. It's amazing to think that something collected centuries ago can still be relevant today.
I know. It's pretty cool.
So now that we've found this lost lectotype, what does it mean for the proposed new species of green anaconda?
Well, remember, the lectotype was found to belong to the northern mitochondrial lineage,
Right.
the same one that the new species is based on.
Okay. Yeah.
And the lectotype is the anchor for that scientific name, Eunectes murinus.
So if scientists decide that this northern lineage really is a distinct species, it gets to keep the original name.
Right.
And the other lineage would need a new name.
Exactly.
Okay.
The authors of that controversial paper, they had already tried to designate a different specimen as the neotype for Eunectes murinus.
Okay.
But now that the original lectotype has been found, their attempt is basically meaningless.
Wow. So their rush to name a new species kind of backfired.
You could say that it really highlights the importance of being thorough and careful in taxonomy.
Yeah.
All those rules and procedures in the International Code of Zoological Nomenclature,
Right.
they're there for a reason. They help to ensure that things are done in a clear and stable way.
Yeah, it seems like skipping steps can really mess things up.
It can.
So this whole situation really shows how important taxonomy is, even if it seems kind of technical or boring to some people.
Right. It's not just about giving names to things, it's about understanding how different organisms are related to each other.
Right, and making sure that our scientific knowledge is accurate.
Exactly. If you get the taxonomy wrong, it can have a big impact on things like conservation. Imagine if we were focusing our conservation efforts on the wrong population because we misidentified a species.
Oh, that would be bad.
It'd be a big problem. So getting the taxonomy right is really important for protecting biodiversity.
Okay, so we've talked about the scientific debate, the naming issues, the rediscovered lectotype. It's been a fascinating deep dive into anaconda taxonomy, but I feel like there's still more to explore.
Oh, there's definitely more.
Where do we go from here?
Well, this controversy has actually opened up a whole new chapter in anaconda research.
Okay.
Now that we have the lectotype back, scientists can go back and look at existing specimens and data with a fresh perspective.
Oh, that's cool.
And there's a push for more sampling, especially in areas where different lineages might come into contact.
So more field work, more genetic analysis, more understanding of where these anacondas live.
Exactly. And it's not just about resolving this particular debate.
Yeah.
This whole thing has sparked broader discussions about the role of traditional knowledge in taxonomy,
Oh, interesting.
the ethics of naming new species, and the need for more collaboration and data sharing among scientists.
It sounds like this is just the beginning of a whole new era of anaconda research.
I think you're right. And this is where things get even more interesting because this whole debate raises a bigger question for taxonomy.
Okay, what's that?
How do we balance traditional knowledge with scientific methods?
Ooh, that's a good one.
Especially when it comes to naming and classifying species, how do we make sure that we're being ethical and respectful of the cultural significance of these animals?
Wow, that's a tough question. It seems like this one controversy has opened up a whole can of worms for the field of taxonomy.
It really has, and it's forcing scientists to confront some uncomfortable truths about the field's history and the biases that can sometimes influence research.
Well, it's certainly been a thought-provoking conversation so far.
It has, and we're not done yet.
It's interesting how this whole thing has really shown that sometimes science isn't about like finding the right answer. It's more about like asking the right questions and challenging like what we think we know.
Absolutely.
And even just accepting that like we don't have all the information yet.
And I think in this case with the anacondas, the controversy has actually been a good thing.
Really?
Yeah, it started these really important conversations about how we do taxonomy now in the 21st century.
Yeah, it's not just like looking in a microscope and measuring things anymore, right?
It's way more complex than that.
Yeah, we've talked about traditional knowledge and the ethics of naming new species and the need for scientists to work together more.
Right, all these different perspectives.
It seems like this one paper really like opened a Pandora's box.
It did, and it's forcing scientists to take a hard look at the history of the field and to think about the biases that can creep into research.
Yeah, and speaking of history, I want to go back to that lectotype for a minute.
Yeah.
It's been so cool to trace its journey from that Dutch pharmacist's collection to the Russian museum.
It's a wild story.
But what does it actually tell us about the green anaconda?
Well, like we talked about before, the lectotype was found to be part of that northern mitochondrial lineage.
Right, the one they were trying to make into a new species.
Exactly. And if that lineage does turn out to be a distinct species, it gets to keep the name Eunectes marinus because the lectotype is linked to that name.
Okay, but what if further research shows that the northern and southern lineages are actually just variations within the same species?
That's totally possible. And if that happens, then the whole debate about the name Eunectes akaima becomes kind of pointless.
So all this drama might be for nothing.
It might, but even if that's the case, I think the controversy was still valuable
How so?
because it showed us that we need to be more rigorous and transparent in our research.
Yeah.
And it forced us to really consider the ethical and cultural aspects of naming new species.
It's like those mistakes in the paper actually helped to move science forward.
Exactly. Sometimes we learn the most from our mistakes.
And this whole thing has definitely renewed interest in anacondas.
Oh, yeah. There's so much more research happening now.
And who knows, maybe there are even more species out there that we haven't discovered yet.
That's what's so exciting about science. There's always something new to find.
Oh, I'm certainly way more fascinated by anacondas than I was before we started this deep dive.
Me too.
And I have a feeling this isn't the last we'll hear about them.
I think you're right.
Well, thanks for joining us on this journey into the world of anaconda taxonomy.
It's been a pleasure.
It's been a wild ride full of twists and turns, but I think we all learned a lot about these amazing creatures.
And about the challenges and the importance of doing good science.
For sure. And for those of you listening who want to learn even more about anacondas and the incredible diversity of reptiles out there, be sure to check out the links in the show notes.
There's a whole world of amazing animals out there just waiting to be explored.
So keep exploring, keep asking questions, and keep diving deep into the wonders of science.
We'll see you next time.
Bye, everyone.